In order for us to understand how muscle contractions take place, it's important to understand a little bit about the nervous system and specifically about the resting membrane potential and action potentials. So this is going to be a very brief and quick introduction to the nervous system. The nervous system is something that you're going to go over in more depth when you get to Bio 50B, but we do need to understand some of the basics in order for us to understand how kind of a motor neuron is going to work and how that's going to then help cause muscle contractions. So we're going to be interested in understanding how motor neurons are going to work. And so this lecture is not going to talk much about motor neurons and how they cause muscle contractions, but it's going to talk about basics. Okay, and so motor neurons are going to be these neurons that are going to kind of end up touching muscle, and it's going to cause muscles to contract. There are other types of neurons, specifically sensory neurons, that you'll learn about in a different course, but we're looking at motor neurons that are going to cause our muscles to contract. So here's the general structure of a neuron. We have kind of a different type of cell shape. The main part of the neuron is going to be called the cell body. So this is kind of what our normal cell would look like. But you can tell that there are lots of different structures on this type of cell. And so we have the cell body, which is going to have the nucleus inside of it. And so it's going to have kind of, your standard organelles that are going to be in there. We're going to have rough endoplasmic reticulums, we're going to have mitochondria and things like that. We're then going to have this large area, kind of this long extended arm, if you can say, which we're going to call the axon. And the axon is going to be the very important structure for our nerve impulses. So what's going to happen is neurons, these cells, are going to receive input from another part of the body, and they're going to receive input on these structures called the dendrites. And the dendrites are going to be kind of all of these little areas that kind of branch out and are going to touch other nerves and receive input. The nerve impulse will then move through the cell body, and then it will go to this area called the axon hillock, which is then going to lead to the axon. Now the axon is going to be kind of the direction that we're going to send our nerve impulse, and we'll talk a little about the nerve impulse in a second. But there are lots of structures on here. There's something called myelin. We have these Schwann cells. We're not going to worry too much about that, right, for this specific class. But what you need to know, the last kind of major structure is going to be this axon terminal, which is where we're then going to have kind of this area where the axon is now going to interact with some other type of structure. It could be another neuron. In our case, we're going to actually talk about how we get to the muscle, and we have kind of this motor end plate, which is then going to interact with the muscle and will be kind of how we are going to conduct neural impulses from nerves to the actual muscles. Now, a little bit about the very end of the neuron. So I kind of drew it at the very end, but the axon terminal is going to have a specialized structure. And one thing that it's going to have in the terminal is going to be these things called synaptic vesicles. And these are just going to be little storage containers for neurotransmitter. Okay, neurotransmitters are going to be specific types of molecules, proteins that are going to interact with kind of the cell that is actually interacting with this neuron kind of axon terminal. And so down here we have this postsynaptic cell. So this could be any type of cell. When we talk about this, it's going to be an actual muscle cell, but this could be anything else depending on kind of where it is and what it's doing. but we're going to be worried mostly about kind of muscles. Okay, and so between the neuron, the previous neuron, so this axon terminal, and the postsynaptic cell, we have this thing called the synaptic cleft, and this is an area between, so we don't have direct contact between the axon terminal and the postsynaptic cell. So there's actually this space, and in this space is where we're going to actually have our neurotransmitters that are going to kind of be released. Sometimes we're going to have other types of chemicals that are released, and they're going to interact with these channels that are on the postsynaptic cell. And depending on, again, what kind of cell you're talking about, it's going to then do some type of response. So in an action potential, and we'll talk a little more about what action potentials are, but I wanted to kind of get the anatomy of the neuron kind of done, is we're going to have an action potential that will be sent down to the axon terminal. It will then cause for the axon terminal to release these synaptic vesicles, and it will do this because there are these voltage-gated calcium channels, so they're in green. These calcium channels are actually going to cause the synaptic vesicles to actually release their neurotransmitter. So action potential comes down because the calcium channel is on the axon terminus. It is going to then react to the voltage change, and the voltage change will then cause the synaptic vesicles to be released, and the neurotransmitter will then be released into the synaptic cleft, which is this area right here between the previous, the axon and the next cell that it's going to interact with. Okay, and then that will cause some type of reaction. So in order for us to understand how action potentials are actually going to be able to be propagated to muscle cells, we are going to need to understand a little bit about electricity. And here are just a couple different important principles that we need to keep in mind, because this will help us understand how action potentials are going to take place and how we can propagate a nerve impulse to a muscle and have the muscle then contract. So the first principle is that opposite charges are attracted to each other. So you're always going to have positive being attracted to negative, and negative is going to be attracted to positive. So they are going to be attracted to each other. So opposites attract, and in this case, like charges would repel each other. Okay. Now, when you have different charges on opposite sides of a membrane, so if we have a membrane, and let's just say this is outside, and this is inside, and inside you have a net negative charge, and so I'm going to draw negative signs, and on the outside you have a net positive charge, so you have kind of these positive. You are going to require energy in order to keep them separated across the membrane, okay? Because their tendency is going to always want to be moving towards, for the positives, towards the negative, and the negatives towards the positive. Now, when the charges move toward each other, energy is liberated from the system. And so if you can create this kind of difference in charge from outside to inside, you have a system that has a lot of potential energy. And this potential energy is going to then be used to propagate a signal from one place to another in the neuron and then to ultimately the muscle. Now, when we have a cell, kind of one of the big things is to know where different ions are. And so this is kind of just a stereotypical cell. And when you have when we're dealing with concentrations, anything that's written very big letters is going to be something that means it has a very high concentration. And anything that is written small means that there is a low concentration. And so in this image, what you're going to see is that there's high levels of potassium inside the cell. There are going to be lower levels of potassium outside the cell. There's high levels of sodium outside the cell and lower levels of sodium inside the cell. We also have chloride. Chloride is going to be high outside and low inside. And then inside the cell, we're going to have organic anions. And anions are just going to be negatively charged molecules. The net charge of the inside of the cell is going to be negative, and the net outside of the cell is going to be positive. Okay, and so the reason why it's negative and positive is because you kind of take into account all the different ions and their charges and see kind of what is your charge. And so it turns out when the cells that we're going to talk about are going to tend to have a positive external extracellular concentration, and internal is going to be negative. Now, when we kind of look at our cells and we have this difference in the charge, we are going to start talking about voltages. And so voltages are going to be a measure of potential energy generated by separated charge. Okay, so if we have on a membrane, we have positive charge on the outside and negative charge on the inside, this is then going to cause kind of a separation in charge, which can be measured in voltage. Now, you tend to measure voltage in something called volts or millivolts. Usually millivolts is what we'll be talking about. And when we talk about voltage, we can talk about the potential difference or the potential of the membrane. And so this is going to be kind of the change or the charge difference between the outside of the cell and the inside of the cell. Okay, the higher the difference, the greater the difference is between the inside and the outside, the higher the voltage is going to be. Now, ion channels are going to be extremely important in all of this. I talked before about how potassium, chloride, sodium, anions, all of those are going to be important. The main two ions that we're mainly going to be concerned with are going to be potassium, which is K+, and sodium, which is Na+. Okay, so there's going to be membrane ion channels that are going to be on the membranes of the neurons. And this is also going to be present on the membranes of the actual muscle cells. Okay, so there are going to be these proteins that are going to allow for membranes to move through, or ions to move through, and they're going to be selective to certain ions. So there are potassium ion channels, which will only allow potassium to pass through. Of the potassium channels, there are two different types. There's one that is a leak channel, which is always open. So regardless of what's happening, if there's an action potential or not, the leak non-gated channel is always open. The second type of channel is called a gated channel, and this one is going to be open or closed depending on other conditions. And mainly this will be, and we'll talk about the gated channels, is when we have an action potential that's going to be coming into the cell. Okay. Now, there are different types of gated channels, and they can fall into these three different categories. So there is chemically gated, voltage gated, or mechanically gated. Okay. So chemically gated are going to be ligand gated, which will only open when they have a specific chemical that binds to them. And so, for example, a neurotransmitter. Okay. When we talk about muscles contraction, we're going to be talking about acetylcholine. And so that would be a neurotransmitter that is going to bind to chemically gated channels. Voltage gated channels will only open when a specific voltage happens. So if there's a change in the membrane potential, if it gets more positive, then you can have these actual channels that will open, but only when the membrane potential has changed because of a change in the voltage. The last is the mechanically gated channel, which opens in response to some type of physical deformation. And these are usually going to have some type of response. Something's going to bind to them that will then cause them to deform and will allow them to open up. So here are some images of those different types. So we have a chemically gated ion channel. So when there is no, in this case, neurotransmitter, the sodium ions cannot move into the inside of the cell. But if we have a neurotransmitter that is bound, then sodium can actually move into the cell. And in this case, this specific channel appears to be a co-transporter because it's going to allow sodium inside and potassium is going to leave. Voltage ion channels can only open when the voltage has actually been changed. And so in this case, in the first image right here, we have a closed channel. And then once there is a change in the voltage, and so you see that, if you can see this, there's positive outside initially and negative inside. And then when we have an action potential that happens, we're actually going to switch we're going to have a negative outside and a positive inside, which then causes an actual conformational change on the protein of this channel, allowing for it to open and allow sodium to come inside. Now, here are two images of kind of ion channels that are going to be important for muscles. The first is going to be a ligand-gated channel that is going to react with acetylcholine. And so we will talk about this more in depth when we talk about muscle contractions. But acetylcholine is going to be a neurotransmitter. It will come in and bind onto the ion channel and allow for, in this case, positive ions to run in. For heart muscle, acetylcholine is actually going to bind to another type of receptor. It's actually going to cause inhibition of muscle contraction, so a little bit different than what we have in the skeletal muscle. Same neurotransmitter, but different effect because of the different types of ion or non-ion channel receptors. All right, when we have voltage-gated channels and they open, ions will move very quickly through them, and they will move along the chemical concentration gradients. And so just like we've talked in previous labs a long time ago, things move from an area of higher concentration to lower concentration. So if there is a lot of sodium outside the cell, not very much sodium inside the cell, and all of a sudden a sodium channel opens, sodium is going to rush into the cell. If there is a lot of potassium inside the cell and not very much potassium outside the cell, the second that an ion gate opens, potassium is going to run out to an area of lower concentration. Now, this also works with charge. So the different ions will move towards the opposite electrical charge. Okay, and so sodium ions are going to be positively charged. The inside of the cell is negatively charged, and so the sodium ions will rush in, which will then cause it's going to be the attraction of the positive to the negative inside, which will then ultimately change the voltage of the entire membrane. So when we have kind of this situation where we have positive and negatives, we can set up a resting membrane potential. And so when you look at an axon of a neuron, when it's at rest, so no actual nerve impulses coming in, you can measure the outside to the inside and see kind of what is your difference in charge. And so on average, the resting membrane potential of a neuron is about negative 70 millivolts. And so the inside is more negative than the outside. So we say the cytoplasmic side of the membrane, so that's going to be inside the cell, is negatively charged relative to the outside. Now, the charge is the voltage is not exactly negative 70 at all times. It can kind of vary from negative 40 to negative 90, but regardless of that, the main thing to understand at resting membrane potential is that we have a polarized situation. And we talked about polar before, so molecules are polar, which means that they have charge on different sides. And so a polarized membrane means that there's a different charge on the two different sides of the membrane. And so, for example, when we look at the neuron on the axon, we're going to have a positive charge on the outside and a negative charge on the inside, which means that that membrane is polarized. Now, the potential, again, is going to be generated by differences in the ion composition of the intracellular fluid and the extracellular fluid. Okay, so what's inside and what's outside the cell. So if we have, again, kind of a net negative charge on the inside and net positive charge on the outside, that is a difference in charge. Okay, and it's also going to be generated by kind of the plasma membrane permeability. So if the plasma membrane is very permeable to sodium ions, for example, then you wouldn't potentially be able to have this charge, because the sodium ions would want to move through into the cell, and then you wouldn't really have this difference in the charge on the different sides of the membrane. So here, again, is just kind of a description of where the ions are. So the extracellular fluid is going to be very high in sodium, and it's going to also have chloride, and so chloride is negative charge, so it kind of is balanced by chloride. The intracellular fluid is going to have a very high concentration of potassium ions, and it's going to be kind of balanced by those anions, and anions are going to be negatively charged molecules, so other things. They could be proteins. Potassium is going to be the most important ion in the role of the membrane potential. So it's going to be very important that there is kind of this difference in where the potassium is going to be. So here's just a summary of key points for the resting membrane potential. So when you have a neuron that is not in a signaling state, so it's resting, you are going to have a voltage across the membrane, and this is called the resting membrane potential. Okay, because it has the potential energy to allow for transmission of a neural impulse. The resting membrane potential is going to be determined by the ions. Specifically, where are the ions, and then the permeability to the ions on the actual membrane. And when you have a resting neuron, there is going to be a concentration gradient across the membrane for sodium and potassium. So those are going to be our two important ions that we are looking at. And each of those ions will move down their concentration gradients if channels are open for them to move. So now that we have a basic understanding of the resting membrane potential, we can now talk about what happens when it changes. And so we have these two terms that are going to describe kind of relative to the resting membrane potential how the voltage is changing. So the first term is going to be called depolarization. And so if you think about it, polar means that there are two different charges on one side and the other. Depolarization is the opposite, where you're actually decreasing the membrane potential. You're moving the voltage difference close to zero and above, and it makes it so that the inside membrane becomes less negative than the resting potential. Okay. Now, when you have depolarization, this is going to kind of increase the probability of producing a nerve impulse. Okay. There's another term called hyperpolarization, and so this is an increase in membrane potential away from zero. So this is when the inside of the membrane becomes more negative, so even more negative than it is at the negative kind of normal resting membrane potential. And when something is hyperpolarized, it's basically decreases the probability of producing an impulse. Okay, so we're not going to talk much about hyperpolarization right now. We are mainly going to talk about depolarization because we're going to talk about action potentials. So now we're going to discuss action potentials. So action potentials are going to be the way that neurons are going to send signals from one area of the body to another. We have neurons throughout our entire body. We're able to communicate from our brain to the tips of our toes, and this is going to allow for long distance neural communication. Now, we only will have action potentials take place inside muscle cells and the axons of neurons. Okay, so you will not see these in like a liver cell or other types. They're going to be only in muscle cells and axons of neurons, which is why we have to talk about action potentials and kind of give you this brief background about the nervous system, because muscle cells are going to be one of the only types other than neurons that are going to actually participate in action potentials. When a neuron sends an action potential, it can be called a nerve impulse, okay? And when action potentials are going to be sent, they're going to involve the opening of voltage-gated channels, because the main thing that's going to happen during an action potential is we're going to go from a polarized membrane to a depolarized membrane, and that's going to change a difference in the charge, the voltage, and you're going to have a bunch of voltage-gated channels open, causing different ions to move in and out of the membrane. So we're now going to go over a brief description of how action potentials are actually going to be generated. So there are 4 main steps, and the first step is at the resting state. So this is when all the gated sodium and potassium channels are closed. We do have leak channels for both sodium and potassium. Those are open, so slow movement of sodium and potassium are allowed to move. Sodium is tending to move into the cell, potassium is going to move out of the cell, but it's very slow and it does not affect the resting membrane potential. If anything, it actually maintains the resting membrane potential so that we have this positive charge on the outside of the cell and a negative charge on the inside. Now, each sodium channel is going to have 2 voltage sensitive gates. We have an activation gate, which is going to be closed at rest, but will open if there's a depolarization, which will allow sodium to enter inside the cell. There is then an inactivation gate that will open at rest and block channel once it's open to prevent more sodium from entering the cell. Okay, so you're going to have both activation and inactivation. There also is going to be a potassium channel, and it has 1 voltage sensitive gate. It's closed at rest and opens slowly with depolarization. So here's the first step of an action potential. So at rest, all gated sodium and potassium channels are closed. And so you can see that we have kind of the activation gate of both the sodium and the potassium. So if you can see right here and right here, there is a block, a physical block between the channel so that sodium and potassium are not able to move. The next step is depolarization. So sodium channels will open. When the sodium channels open, that will allow for sodium to rush inside of the cell. Now remember, sodium is going to move into the cell because there's not as much sodium inside, and also the charge of the inside of the cell is very negative, and therefore positives are going to be attracted to that negative. The sodium activation and inactivation gates will open at this depolarization, and the sodium influx causes even more depolarization, which opens even more sodium channels. And this, as a result, makes the inside of the cell or the intercellular fluid becomes less negative. Okay, and so becoming less negative is that depolarization. When we get to the depolarization of about negative 55 to negative 450 millivolts, there's this positive feedback that causes the opening of all the sodium channels. Okay, and so this results in a large action potential that will spike, and the membrane potential polarity actually jumps to positive 30 millivolts. So it actually goes way positive. So there's a lot of positive going inside of the cell. Okay, so this is shown on this image. So we have this part of the sodium channel is going to be in a different conformation, allowing for the sodium to enter the cell. Then we have repolarization. So this is going to repolarize or make the difference between the inside and the outside back to negative. Okay? So the sodium channels are going to inactivate. You have the sodium channel inactivation gates will close. The membrane permeability to sodium will decline to the resting state. And so there will still be sodium leak channels that are allowing sodium to come in, but you have no rush of sodium coming in at this point. This causes the action potential to stop rising because we're going to stop having a rush of positive entering the actual cell. Now, at this point, we have the voltage-gated potassium channels are going to open. And at this point, we now have kind of a very positive charge on the inside of the cell, and there's now a relative negative charge outside the cell, and so the potassium is going to exit down its electrochemical gradient. The electrochemical gradient, again, means that it's going to go down kind of the electrical, so it's going from positive to negative. It's attracted to the negative outside now, but also there's less potassium outside the cell than there is inside, and so it's going to tend to go down its concentration gradient. Now, when you start to have positive charge leave the inside of the cell, that's then going to kind of take the membrane back to its polarized situation, which we call repolarization. So the membrane is going to return to resting membrane potential. So here we have if you can see, this inactivation gate is going to close. It's going to cause the channel to be closed to sodium, so sodium can no longer rush into the cell. And we're going to have our potassium gate open, and so potassium is now going to be able to leave the cell. Now, when we have the potassium leaving, it causes something called hyperpolarization, which is when the potassium is actually going to leave at such a high rate that it actually causes the inside of the cell to become even more negative than it was prior to the action potential. So some of the potassium channels will remain open and the sodium channels will start to reset. Okay, so we will have kind of an excess of potassium leaving the cell, which makes the inside membrane become more negative than before, and that causes the hyperpolarization, so that means more negative, and it will actually dip below kind of the normal resting membrane potential, but it will eventually get back to resting membrane potential. At this point, the sodium channels again begin to reset. So when we're talking about the sodium channels resetting, you're going to notice that kind of this part of the protein channel is now back so that we have this block in the middle, and this inactive gated chain part is actually off of the channel, so that it's only at this point going to be blocked by this voltage gated activation channel. We still have potassium leaving the cell at this point, causing a hyperpolarization. Now, when this happens in an axon, what happens is you have a propagation of the action potential. So what we just saw was in one location of the membrane. But when you have an action potential, it actually will be propagated, which means to move down the actual axon. Okay, so it'll be transmitted from the origin down the entire axon length toward the terminus, which will be the axon terminal. The sodium influx through the voltage gates in one membrane area will cause other areas right next to it to also start to open, which then leads to kind of this propagation or wave of depolarization down the axon. Okay, so you kind of have the rush of sodium going inside, so sodium's entering, and then you have, so this is becoming positive, which then will open more channels, and so you just have a propagation of sodium entering down the axon terminal. Once the action potential hits a specific threshold, and again, we're not going to talk about a lot of that right now in this part of the class, but you'll learn about this in other parts. But if you hit a certain level and the action potential reaches threshold, it will then be self-propagating. So it'll kind of go through its entire cycle until the kind of nerve impulse is gone. Now, there are going to be different types of axons. Some of them are going to be called non-myelinated, and other ones are going to be called myelinated. they're going to differ in how they actually are going to move the action potential down the axon, but that's not really important for us to understand right now. But the main thing is that you will have kind of a time where the sodium channels are going to be closer to the action potential origin, are going to be still inactivated, so no new action potential can be generated there. So there's this refractory period that makes it so that you can't send nerve impulses right after each other. There has to be a little bit of a gap between them, because we go through this inactivation stage of the sodium channels where they have to then go back to kind of ready in their resting position.